let's learn about monoclonal antibodies. There's something quite significant that we need to be aware of here, and that is what the word monoclonal antibodies actually means. Mono means one. Okay? Clonal means lots of. An antibody means antibodies that are produced as part of the immune response by white blood cells. And they attack invading pathogens. Okay, a pathogen is a microorganism that can cause a disease. Okay. They destroy pathogens. And pathogens are disease causing microorganisms. Okay. Now, something quite special about these pathogens, they have on their surface molecules called antigens. Okay. So these pathogens, I'm going to draw it as a P like this, has on its surface very specific molecules called antigens. Okay. Now, if that pathogen was to get into our body and actually break through, the main defense against microorganisms, against pathogens in our body is our skin. That's the main physical barrier. We have other things like enzymes in our tears and saliva and nasal hairs. We have all these other things that help protect us from pathogens. But if that pathogen does get into our body, past our skin, and into our blood, then something quite unique happens. We initiate an immune response. So this is skin, and this skin, the molecules of skin, they also have a variety of very unique antigens on them. Antigens are protein molecules, okay, with very specific shapes. But you know the antigens on our skin, our body knows that those antigens belong to us, and these antigens are recognized as self, belongs to me, belongs to us, it's fine. When that pathogen, though, gets into our body, when that pathogen gets into our body, the antigens of the pathogen are recognized as, oh, don't belong to us. So these, because it's a foreign particle, it doesn't belong to our body, these antigens are recognized as not belonging to us, as non-self by white blood cells. And the first white blood cell that recognizes this pathogen as not belonging to us is called a phagocyte. So as that pathogen enters our blood, the first thing that happens is this phagocyte does this. It recognizes those antigens and it moves towards the pathogen like this. Okay, so phagocyte moves towards pathogen because antigens are recognized as non-self. Okay. When the phagocyte goes towards a pathogen because it knows it doesn't belong to the body, it does something else. It does something kind of special. It changes its shape to surround so this is still our phagocyte. it's moved towards a pathogen because it recognizes antigens as non-self, and it changes its shape. Okay, it changes shape, and this word's always on the mark scheme, to engulf pathogen. Inside the cytoplasm of this phagocyte, we have a little vesicle containing enzymes. And this vesicle containing enzymes now moves towards the membrane. It joins with that membrane like this, which means then the enzymes can be released from the vesicle onto the pathogen, and these enzymes destroy the pathogen. Okay. At the same time, these antigens now, they've got a very specific shape. The body recognizes the shape of these antigens as being not belonging to us, not belonging to us. So what other white blood cells do, and these white blood cells are called lymphocytes, beta or B lymphocytes, they make antibodies that are specific to the shape of the pathogen's antigens. And that's a great idea, because this pathogen is a microorganism. It's going to make lots and lots of copies of itself, right? We don't want that to happen. Well, that is going to happen. When the pathogen has made lots of copies of itself and cells are struggling, then you have symptoms like we feel unwell. But then when our immune system starts to make the antibodies that are needed, then the antibodies bind to the antigens. They match in shape. They're complementary in shape. Destroys pathogen. Okay. That is how antibodies are made, specific in shape to a pathogen. Okay. It's important that we understand that before I go on to monoclonal antibodies. Okay. So that's just me telling you how antibodies are made in our immune system. Well, monoclonal antibodies means lots of antibodies of one type. Okay. Why would we want to make lots of antibodies that are one shape? Well, have a think about this. Have a think about this. This is a cancer cell. This cancer cell has very specific molecules on its surface, protein molecules with a very specific shape. Okay. What if we could make a molecule that matched the shape of the cancer cells? So when we put this molecule in a human with the disease of cancer, then we could make this antibody bind to these cancer cells. What if on this antibody we added an enzyme that could then destroy that cancer cell? That would be a great idea. In order to target the right cells so that the enzyme destroys the right cells, we need a molecule that's going to take our medicine there. Monoclonal antibodies can be used for this. Okay? Monoclonal antibodies, making an antibody that has a specific shape. In essence, what we're doing is just making a molecule that's a specific shape. What if we want to help someone who is struggling with a disease? What if we want to help someone who has got lots of these in their body? Or what if we want to help someone who can't make the antibodies because they've got an illness or something like that? Maybe we'd want to make lots of these antibodies. Okay. So making monoclonal antibodies is a very kind of important thing. I'm just going to go with the second example here. How can we make lots of antibodies that's going to help someone with this illness? Imagine getting an injection of all these antibodies. That would be a dream, wouldn't it? You could give all those antibodies to a patient, and that would do the work of killing the pathogen for them. So how can we make lots of antibodies like this? Well, first and foremost, we take the pathogen with the antibody, with the antigen on it that we want to match the antibody to. So we take the desired pathogen that we want to be able to protect against, but we kill the pathogen. So if it's a bacteria, we just kill it, but we keep the structure of it. So we just destroy the DNA, so it can't replicate or do anything, but we keep the design of the outside structure. Okay? And we put that into an animal, like a rabbit. Okay? Now, when we put that pathogen into that rabbit or into that animal, small animal, in the laboratory, if we put that pathogen in here, the white blood cells, they're going to start making these antibodies. They're going to start that immune response. They're going to be making the antibody that is the exact design, matching in shape to the one that we want to make. They're going to make loads of it. That's a great idea for the rabbit, but how are we going to get the antibodies out of the rabbit to our patient? It's a bit tricky, that bit. So instead, what we do is we take the white blood cells from the rabbit. We take some other cells that grow and replicate fast, because now we've got the right design of antibody. Now we want to make loads and loads and loads of them. So we take a cell that replicates quickly. In the laboratory, we take a cancer cell. These cancer cells, they replicate quickly. And we put the white blood cell and the cancer cell together. And we make a cell called a hybridoma, which can now multiply fast, make lots of copies of itself, and therefore make lots and lots of antibody. We've got now lots of cells making the antibody that we want. From this one hybridoma cell, we let it undergo mitosis, cell division, it doubles and divides, and makes lots and lots and lots of antibodies. What we then do is we just take the antibodies and we put them into a medication that can then be used for a patient. Okay.